Okay, well, hello, everyone. My name is Michelle Olguin, and these are my other team members, Diana Palman and Yarismel Rodriguez. And our project is on the effect of microgravity on biofilm formation by E. coli on polystyrene particles. And to begin with, bacterial biofilms are communities of bacteria surrounded with a slime-like substance that creates a protective shield and makes them resistant to a variety of environmental factors, including antibiotics. The objective of our study is to determine if biofilm formation by E. coli K12 on polystyrene particles is influenced under microgravity conditions. Our hypothesis was that microgravity will influence the ability of E. coli to form biofilms on these polystyrene particles.
Our materials were 500 micrometers diameter of polystyrene beads, E. coli K12 strain, Luria bertani, which is the LB broth, 0.85 sterile saline solution, pipettes, and the Hitachi 1000 scanning electron microscope. Our E. coli K12 was cultured at 37 degrees Celsius for 24 hours and diluted to a final concentration of 1.4 times 10 to the 8th colony-forming units per milliliter. Our experimental samples consisted of 60 polystyrene beads, combined with 90 micrometers of 0.85 saline mixed with 125 micrometers of E. coli suspension. Our bicycle experiments were then set up for microgravity and ground control conditions. Our microgravity and ground control conditions were, you could say, locked down in a dark environment for 15 days, 17 hours. Our samples were then observed in the scanning electron microscope, and if biofilms were located, they were measured by length, width, and thickness.
These are examples of our ground control beads. You can actually see here on the left 3 different beads at 50 magnification. The bead here on the right is actually the bead in the middle at a higher magnification. These are 2 different samples of biofilms. This one here on the left is at 6,000 magnification. Notice the little webbing. This is the biofilm formation. And here is at 10,000 magnification. Additional of our ground control beads, we also did in a cross section to give us a better idea of the thickness, the width, and the length. Here on the left, you can see where we did measurements, and this is at 2,500 magnification, and on the left is at 5,000 magnification. This is another example of our ground control bead at 5,000 magnification. We zoomed in and were able to identify that we had individual cells of E. coli. We also had to determine a way to visualize that we did have E. coli within the biofilms by washing them very carefully with decolorizer. And this is another example of one at 5,000 magnification. As you can see now, you can visually see the cells. When our microgravity sample came back, we began testing to make sure we had viable E. coli in each sample. On the left is our ground control. These were both grown on EMB Media, which is specific to showing E. coli in a green metallic sheen. On the left is our microgravity, or excuse me, on the right is our microgravity sample with no visual growth of E. coli. Additional tests done with our microgravity experiment, we used a TSA media and a blood agar. Using an identification system, we found that the bacteria we received back that overgrew our E. coli was Sphingomonas passimobilius, and we had a 99.99% probability of its correct identification. In conclusion, our results, we had at least 60% of our surface of our ground control beads covered by biofilms. Biofilms size range from 11.6 to 17 micrometers up to 112 to 72.3 micrometers. Biofilm thickness ranged from 267 nanometers to 6.8 18 micrometers. We were unable to recover any E. coli from samples exposed to microgravity to determine biofilm characteristics at this time. However, we are going to be able to refly our experiment on Atlantis.
We would like to do some acknowledgements. Jose Mendoza, Gloria Anchondo, and Dr. Jeff Goldstein in the ECC program, and also John Casanto in the ITA for letting us refly our experiments. The project was supported by the Texas Space Grant Consortium, the Foundation of EPCC, the MISIP grants, the Rice grant, and the El Paso Community College President's Office. And just to know, we're from El Paso Community College and Trans-Mountain Early College High School from El Paso, Texas. Any questions?
Were there any benefits for this experiment?
Right now, our initial observations were to see the difference between how the biofilms would form under gravity conditions and microgravity conditions. As is already stated, that biofilms create a resistance towards disinfectants and antibiotics.
Thank you.
How were you able to get your project to refly on the Atlantis?
Well, as we did multiple testing, we were not able to find any E. coli. We just determined that another bacteria had overgrown it. But the other part of our sample were the polystyrene particles, or little beads. If you have a pen in your hand and you look at the tip, that's how big they are. And they were clear in size. Unfortunately, we were unable to receive any back. And to the untrained eye, an air bubble can look like a bead. So I guess they wanted to give us a go again.
Thank you again for an excellent talk.
Someone else to hold that besides me?